So we've now talked about how evolution happens broadly. We've talked specifically about population genetics and how evolution happens at the level of the population across generations, meaning shifts in allele frequency and characteristic shifts in a population. We've talked about how that can lead to speciation, where two groups diverge to the point where they can no longer exchange DNA, and they are independent species. So we know that one species can lead to other species. But where did that first species come from? Well, today, we're going to take a look at what may have happened in the beginning. We talked in Bio 131 about cell theory, which essentially says that all organisms on Earth that are living are made of cells or are single-celled creatures, and that life can only come from other life. So cells can only come from other cells. And that is true almost universally, the only exception being the first cells, right? The first cells could not have come from other cells. So how did those cells come about? Well, we don't know exactly because we weren't there and it was a long time ago, so evidence is relatively scant, but we do have some ideas that are based on what we know now of the processes of cell division. So first, you need the molecules of life. And we need abiotic synthesis of the molecules of life. So organic molecules, things like amino acids and nucleic acids and carbohydrates like glucose, those things, when we find them in the wild, we typically find them produced by living organisms, right? Those are evidence of life. So we have to have abiotic synthesis of those molecules, meaning synthesis not by a living organism. And then those have to join into larger macromolecules. So those amino acids have to join together into proteins, right? Those nucleic acids have to join together into larger nucleic acids, right? Those monosaccharides have to join together into polysaccharides. And then those things have to get packaged into a phospholipid membrane, and we call these things protocells. And then we also have to have had an origin of self-replicating molecules, right? Like nucleic acids, which can copy themselves. Now, as we start to think about how this might have happened, we have to go back to what the Earth was like as it began. So that it was a little over 4.5 billion years ago when the Earth was formed, along with the rest of the solar system. And for the first, 0.6, 0.7 billion years, it was a violent place to be. The Earth was bombarded by rocks, and ice probably vaporized into gas, and there were not actual seas forming at that point in time. It was a pretty nasty place. It was not very hospitable to the evolution of life. Additionally, the early atmosphere, in addition to that water vapor that we talked about, probably had a lot of chemicals that came from violent volcanic eruptions, things like nitrogen, nitrogen oxides, carbon dioxide, methane, ammonia, hydrogen, hydrogen sulfide, things like that, but no oxygen. And oxygen is not very common on its own in the universe, and it was not present in any appreciable amount in the Earth's atmosphere early on. That didn't happen until there were organisms that could make that oxygen through photosynthesis. So we know that the environment in the atmosphere of early Earth was different than it is today. And as early as the 20s, scientists were recognizing this and were hypothesizing that it was a reducing environment rather than an oxidizing environment, if you remember that flow of electrons. And it wasn't an oxidizing environment, again, because there was no oxygen. And a few decades later, Stanley Miller and Harold Urey conducted what became one of the more famous experiments in the history of science. And we'll talk about it in a moment, but essentially they recreated the atmosphere that they had hypothesized for early Earth. They added in some energy in the form of lightning, right? They had some electric shocks, and they found that organic molecules like amino acids just popped up, right? They formed on their own in the conditions of that early universe. Now, their hypothesis was sort of the primordial soup hypothesis, the idea that there was some warm pond where life occurred, right, during the early Earth. And this is consistent with what people all the way back to Darwin had sort of hypothesized. And that may be the case. There are other people now that suggest that it's more likely that it could have happened near deep sea vents on the bottom of the ocean. And there are some people that suggest that life or the materials that led to life could have arrived via an asteroid from outer space. Although, again, if you're looking for the origin of life, that doesn't explain the origin of life. It just pushes the question back to Mars or somewhere else in the universe that would have launched a rock full of microbes to Earth. And whether this occurred in a deep sea hydrothermal vent or at the surface in a warm pond, essentially the same set of things would have occurred. Right? You have the same set of compounds that you have in the early Earth's atmosphere, nitrogen-containing compounds like ammonia and such, and you get some sort of energy either from the vent or from lightning, and you wind up getting these simple biological molecules synthesized abiotically, right? And Miller and Urey's experiment, which showed this, has been reproduced many, many times in slightly different circumstances over the years, and it holds up. Now, the next thing that needs to occur is those monomers have to join together to form larger molecules. So polymers need to arise from monomers. And it turns out this can happen if you just leave them together for long enough. But it also happens more quickly on hot sand, clay, or rock. Now, once you have macromolecules, the next step in producing cells is a cell membrane. You have to have something that separates the inside of the cell from the outside and helps to isolate that chemical environment, allow it to be different from what's inside, right, and relatively consistent. And it turns out that what we have composing our plasma membranes are phospholipid bilayers. And phospholipids are among these abiotically synthesized organic molecules. And if you put them together in water, remember they have one end that's hydrophobic, right? The big long fatty acid tail, and the other end is a little short hydrophilic phosphate. And if you put those together, they spontaneously, because this is the most stable way for them to exist, form a bilayer. And in fact, not just a bilayer, but a sphere, right? Because then those edges don't have to be exposed to the water. And so they naturally just form these spheres, these vesicles that have a membrane-like structure. And when these are floating around with these abiotically synthesized macromolecules, you can get what kind of look like cells, right? In that they have a plasma membrane and organic stuff on the inside. And again, this can happen spontaneously, but clay or hot clay can catalyze the process, meaning make it happen a whole lot more quickly. And these vesicles can exhibit simple reproduction. They can break and divide in half, right? They also have simple metabolism because you can have things like simple enzymes in these organic molecules, and they maintain this internal chemical environment via this plasma membrane. And right here you can see the difference in rate between just the precursor molecules and the precursor molecules on clay, right, in terms of how quickly, right, in terms of, these are just minutes, right, how quickly vesicles spontaneously appear. It happens on its own, but it happens way faster with this clay. And at this point, you're pretty close to what we might consider a cell. We call these protocells. What you need is some way to carry the information consistently from one generation to the next so that you can have essentially proper evolution, right? Where you have not only diversity between these proto-organisms, but heritable diverse characteristics. And that brings us to nucleic acids. Now we know from Bio 131 that nucleic acids are remarkably good at copying themselves with some level of fidelity, right, with few mistakes. And that's because of the complementary nature, right? If I line up some nucleic acids over here, and it's ATTGC, right? You know that the other side must be TAACG, or, whatever the opposite of this was, right? Where if you have an A over here, it's always got to be a T over here. If you have a C over here, it's always got to be a G over here, right? That's the way it's stable. And so nucleic acids are particularly good at copying themselves and doing it with fidelity. But DNA, while it's amazing, it's carrying information in a stable way and coding for proteins, right? It can't catalyze reactions like copying itself without the help of protein enzymes. But protein enzymes require DNA to code for them. So, what was first, the chicken or the egg, the DNA or the protein? They both require each other in order to exist and function. And the answer probably is that the first nucleic acid, the first molecule of inheritance, was not DNA, it was RNA. RNA can do a lot of the same things that DNA does. It has that complementary nature. It's not quite as stable as a double-stranded DNA molecule. It's single-stranded. But because it's single-stranded, it can also fold in interesting ways and make interesting shapes, kind of like proteins can do. And that allows it to form enzymatic functions, right? And we know this because there are still RNA enzymes in your cells today. We call them ribozymes, right? Your ribosomes are enzymes that are made at least partly of RNA, and there's some other example of RNA enzymes as well. So RNA can fold into weird shapes and perform functions like proteins can, if not quite as effectively as proteins. And they can also carry information, if not quite as stably as DNA. So they were probably the first do-it-all, one-stop shop molecule of inheritance in the original early cells. And then after a relatively short amount of time, evolutionarily speaking, they may have begun coding for proteins that were far better at doing those jobs, right? And they would have been more successful if they coded for proteins. You may have had RNA that through some mutation began to be double-stranded and became DNA, which could then code for mRNA, which could then code for proteins. And you have a much more stable molecule carrying the information to the point where today, right, everything has DNA as its information molecule, right, which indicates that while there may have been lots of types of proto-life at the very beginning, our last common ancestor, meaning whatever cell it was that led to everything that exists today, was probably a cell that had DNA as its genetic material and produced proteins for enzymes. So, to review, abiotic synthesis of small organic molecules was able to happen in the atmosphere of the early Earth, which did not have oxygen and did have a lot of molecules that were full of nitrogen. And as proved by Miller and Urey, when given some energy, spontaneously, abiotically, without life, produce these small organic molecules. When you put these small organic molecules together, they tend to link up, especially when they're on clay or particularly hot clay. Some of these, the hydrophobic ones, form these plasma membranes, these bilayers, which tend to form spontaneously, especially around hot clay, and can form around these macromolecules. And some of those new abiotically synthesized organic molecules can self-replicate. Probably RNA was the first self-replicating molecule, and now you have these early cells, right, that can evolve and compete until at some point our last common ancestor with all life on Earth evolved that had DNA as its genetic material and proteins as stable, effective enzymes.